Hello, my name is Isabel Weeks. Over the summer, I've been researching the effects of far red and near UV light on plant growth. Far red and near UV light are the two extremes on the spectrum of light that is detectable by plants. For my research, I utilized LEDs, also known as light-emitting diodes. LEDs produce a narrow bandwidth light, and over the last decade, the use of LEDs as a primary lighting source for food production has gradually increased. As a result of this increase, LEDs have been developed on a wide spectrum of light ranging from far red to near UV. Despite this increased use of LEDs in agricultural systems, there's a lack of information on how these two extremes of the spectrum affect crop production. The aim of this study was to determine the effects of far red and near UV light on plant growth, mineral uptake, and phytochemical biosynthesis utilizing scarlet kale. Phytochemical biosynthesis simply refers to the production of secondary metabolites and antioxidants in plants. The minerals and secondary metabolites contained in plants play an important role in human health through our diet. The first step in my research was to sow the kale seeds in a 128 plug tray and then grow them for 5 weeks. The plants were then transplanted to 4 36-well trays and moved under 4 distinct light treatments. These treatments took place in a growth chamber with controlled temperature and humidity. LED modules generating distinct spectrums of near UV, blue, green, red, and far red light were used. The pictures below show the 4 light treatments, and the photosynthetic photon flux density was 150 micromoles per second meter squared for each treatment. Red, green, and blue light were utilized in all the treatments, while the use of far red and near UV light was varied between the treatments. The plants were grown under these respective treatments for 5 weeks before they were harvested. Prior to the harvest, the average plant height was recorded, and after the harvest, the fresh weight was recorded. An inductively coupled plasma analysis, also known as ICP, was also performed to examine the mineral uptake for each treatment. All of the collected data was analyzed using JUMP. Moving on to our results, the first graph shows the average height of the plants for each treatment. A pairwise comparison showed that either far red and near UV light both affect plant height in combination, or they affect it separately. The next graph shows the fresh weight of the kale plants for the 4 different treatments. Statistical analysis showed that there was no significant difference in fresh weight between the treatments. The first graph on the right-hand side is the ICP analysis of the macro elements calcium, potassium, magnesium, and phosphorus. This analysis showed that there was no difference for the uptake of these elements between the treatments. The second graph below that is for the micro elements boron, copper, iron, manganese, and zinc. This showed that there was a significant difference between the treatments for boron and manganese uptake. It was found that exposure to far red light increased the boron uptake, and that exposure to near UV light increased the manganese uptake. The last figure shows representative roots for each treatment. As you can see in this image, exposure to far red light decreased the root capacity of the plants. The next step for this research would be to perform a non-targeted metabolic analysis that would investigate the roles of near UV and far red on a molecular level. I'd like to thank the Department of Soil Sciences here at West Virginia University, as well as the Sugargrove program, for providing me with this opportunity. Thank you all for your time. Please leave any comments and questions you have below, and I'll get back to you.